Hi biologists. Let's start by looking at the learning objectives for this lesson. At the end of this section, and following the biology syllabus, take note that this material is for the higher level biology only. You should be able to use the Punnett square technique to study the inheritance to the second filial generation, F2, of two unlinked traits. What does this actually mean? What are we trying to learn? Well, we have to be able to use the Punnett square technique to study the inheritance of two unlinked characteristics or traits to the second generation or to the second filial F2 generation. Strictly speaking, to fully understand the concepts here, I would advise you to watch my video on Mendel's Dihybrid cross. For those of you who would like to work ahead, I have included the main points needed for understanding here. We now need to be able to explain Mendels Second Law based on chromosomes. Don't forget that a gene controls a characteristic, and each gene has two different forms or two different alleles. So do you recall that we have a gene for height in pea plants, and the gene for height has two different forms. One allele is big T, which is for tallness, and the other allele is small t, which is the gene or the allele for smallness. One allele is dominant and it masks the effect, or it bullies the effect of the other allele, which is recessive. Two alleles, or two forms of the gene, are needed to control the outward appearance or the expression of the gene. So to be tall or to be small, you have to have the two alleles. Homozygous means that the two alleles are the same. So if you are tall, your genotype or the type of genes you would have are represented by big T. And if you are small, your genotype or your genes would be represented by small t and small t. Heterozygous means that the two alleles are different from one another. So if you're a pea plant and you are tall and you are heterozygous, then you are big T, small t. Don't forget, of course, the word homo means the same, whereas hetero means different. Now, I find it easier to understand chromosome behavior in gamete formation if you imagine you have 4 sweets in your hand, 2 blue ones and 2 green ones. They happen to have letters written on them, rather like M&Ms. Now, when you write letters in genetic crosses, you must make sure that you distinguish the letters one from another, that you distinguish the capital letters from the small letters. That is why I have the letter Y underlined here to show that it is a small letter Y and to distinguish it from capital letter Y. Now back to the sweets. Imagine you've been told to half them with your brother or sister. What are your options? Well, you might start with 4 sweets, but you're going to have half them, and your sibling is going to end up with only 2. So what are your options? Well, they're rather fussy, so they only want 1 blue sweet and 1 green one. So what combinations can you give them the sweets in? Well, you could give them the blue sweet with the capital letter T with one of the green ones with the capital letter Y. Your other option would be to give the blue sweet with the capital letter T and the green sweet with the small y. Another option would be to give them the blue sweet with the small letter T and the green sweet with the big letter Y. And your last option would be to give them a blue sweet with the small letter T and a green sweet with the small letter Y. There are your 4 options. Of course, this is representing chromosomes. So therefore, this is a heterozygous parent. A genotype, or the type of genes that it has, is big T, big Y, small y. We are going to form gametes. We're going to half the number of chromosomes. So when you're halving the number of chromosomes, we end up with 4 options. We end up with big T, big Y, big T, small t, big Y, small t, small y. Now, this is the understanding of chromosome behavior in gamete formation. But we do have a trick that we can make use of when we're doing genetic crosses. I call this the arrow trick. Basically, if I take my big letter T and draw an arrow out into big Y, I can create a gamete that contains big T, big Y. I can draw another arrow out of the big T and go into the second letter of the Y's, and then I will create the gamete big T, small y. I can draw an arrow out of the small t into the first of the Y's, and then I will create the gamete small t, big Y, and I can draw an arrow out of the small t into the small y and create the gamete small t, small y. So using the arrow trick, you can see that I end up with the same 4 options. And we make use of that trick when we're doing our genetic sums, as I call them, or your genetic crosses. Now let's represent this by means of a chromosome diagram. Here we have a pair of homologous chromosomes in blue in charge of the characteristic of height in the pea plant. We have 2 alleles for the gene for height. We have big T representing tallness and small t, the allele for smallness. We have a pair of homologous chromosomes here in green for the characteristic of pea colour. The gene for pea colour is represented by 2 different forms, big Y for yellow and the allele small y for green. Now this parent is heterozygous, so we have the possibility of getting 4 gametes. Remember our arrow trick? The first gamete will contain a big T with a big Y. The next gamete will contain a big T with a small y. The next gamete will contain a small t and a big Y, and the last gamete, the last possibility, is that it will contain a small t and a small y. Let's go. Let's cross a heterozygous organism with a homozygous recessive one. This is a dihybrid cross because it involves 2 characteristics. The characteristic of height, the contrasting traits being tall and small, and the characteristic of pea colour, the contrasting traits being yellow and green. As with all dihybrid crosses and all genetic crosses, make sure that you write in the key words shown here in green. I have seen these key words worth 1% or 3 marks of the exam. Make sure also that you make your capital letters and your small letters look completely different from each other. Now, if we take our tall, yellow, heterozygous parent, the genotype will be big T, small t, big Y, small y. Crossing with the homozygous recessive parent, the genotype of which is small t, small y, small y. In the past, in my day, this would have been called a back cross or a test cross. Nowadays, it's called crossing a heterozygous organism with a homozygous recessive, as we can see above. Now, when the heterozygous parent is going to form gametes using our arrow trick that we've just revised, the 4 possible gametes are big T, big Y, big T, small t, big Y, and small t, small y. 4 possible gametes in the other parent, small t, small y, small t, small y. Using the arrow trick for the sake of completion, we can see that all 4 possibilities are going to be the same here. What gametes from the parent on the left will fertilize what gametes from the parent on the right? So to see fertilization, we're going to make use of the Punnett square technique. And in the Punnett square, we will put 1 set of gametes across the top of the square. We put the other set of gametes down the side. It doesn't matter what gametes we put where. The Punnett square will show us the results of fertilization. The offspring we will get when the gamete small t, small y fertilizes big T, big Y is big T, small t, big Y, small y. Working our way across the table, we will see the different outcomes. For the sake of completion, we will fill the entire table, and when we do this, we will discover that we have 4 different types of offsprings in a ratio of 4:4:4:4. So there'll be 4 tall and yellow ones, 4 tall and green ones, 4 small and yellow ones, and 4 small and green ones. From a mathematical point of view, that is a ratio of 1:1:1:1. So in a nutshell, when you carry out a dihybrid cross with a heterozygous parent and a homozygous recessive organism, you will get the ratios of 1:1:1:1. Now, just before we leave that point, for those of you who are sharp-eyed amongst us, you might notice that we have 4 different types of offspring. When you look at the heterozygous parent, we see that the heterozygous parent is heterozygote, so it's carrying a big T and a small t. So in the offspring, we see offspring with the characteristic of tallness, and we see smallness in the offspring as well. The heterozygous parent for yellow is big Y, small y. So again, we see yellow in the offspring and we see green in the offspring. In the back of your head, as a little trick, not terribly scientific, but as a little trick, you have 4 different letters in the genotype of the heterozygous parent. Therefore, we have 4 different genotypes for the offspring. As we've just said, the expected ratio due to Mendel's second law of independent assortment, when you cross a heterozygous parent and a homozygous recessive parent, is a ratio of 1:1:1:1. Now, why is that the case? Well, the reasons are exactly the same as before. We get this ratio because there were 4 possible gametes from the heterozygous parent. We get the ratio because the genes for both characteristics are not linked. So the 2 alleles for tallness, big T and small t, are on 2 separate chromosomes, and the 2 alleles for yellow, big Y and small y, are on 2 separate chromosomes. We also get the 1:1 ratio because the law of independent assortment occurred. In other words, the pairs of alleles, the big T and the small t, separate. They go into separate gametes entirely independently of the other pair of alleles. The Ys will separate from each other and go into gametes separately, totally independently of what the T's do. Now, for those of you able for a little bit more, we have some added detail. Strictly speaking, this isn't entirely on the syllabus, but it does deserve to be looked at. Let's have another look at this idea of a test cross. This is a method in the past that was used to determine an unknown genotype. What do I mean by that? Well, a test cross is a cross between an organism with an unknown genotype and a homozygous recessive organism. Let's have a look at this again. When you actually look at the cross that we've just carried out, the parent's phenotype is tall and yellow. But when you think about it, there are 4 possible genotypes for that phenotype. You could be, or could have, big T small t, big Y small y, which is the cross we've already carried out. You could have a big T small t, big Y, big Y. You could have a cross, big T, big Y, small y as a possibility. And you have the 4th possibility of big T, big Y, big Y. Now, the trick here is that when you look at this parent, you can see the phenotype, you can see that it's tall and yellow, but you cannot see, and you won't necessarily know, the genotype. As we've just said, it could be one of these 4 possibilities. So when you think about it, if I take any of these parents, cross them, do a back cross, cross them with a homozygous recessive organism, look at the offspring, I might be able to work backwards and figure out what the parent's genotype was in the first place. So let's work through these crosses and see what we get. So the parent, big T, big Y, working through our normal rules, using our arrow trick, we can see that we have 4 possible gametes from the parent here on the left, 4 possible gametes here on the right. Using our Punnett square technique, we can see the possible offspring from such a cross. We can see that in the Punnett square, 8 offspring will be tall and yellow, 8 offspring will be small and yellow. Or from a mathematical point of view, the ratio would be 1:1. We should have anticipated that anyway using our little trick that we tucked in the back of our head earlier on, because when you look at the genotype of the parent, you can notice that the genotype of the parent has a big T and a small t. So in our little trick, this is why tallness is showing up in the offspring and why smallness is showing up in the offspring. When we look at the parent, the genotype for yellow is big Y. There's no gene for greenness here, so the results are bearing that out, because in the offspring there's no sign of a green P, they're only all yellow. So if you like, the ratio is 1:1 for tall and yellow, small and yellow, or 50-50 tall yellow guys and small yellow guys. If we look at the next cross, we can see maybe the same thing. Following the usual rules, following the arrow trick, seeing that you have 4 possible gametes in the parent on the left, making use of the Punnett square technique, filling out the table for the sake of completeness, we see that in the Punnett square you get 8 tall and yellow offspring, 8 tall and green offspring. Or from a mathematical point of view, you get a ratio of 1:1. Working backwards, we should have anticipated that, because if you look at our trick that we're keeping in the back of our head, the genotype for tallness in this parent is big T, so there should be no sign of smallness in the offspring. And that is true. All the offspring are tall. Because half the offspring are yellow and the other half are green, then we should be anticipating, if you work backwards, that the genotype for yellowness in that parent was big Y, small y. Let's look at the next cross. Again, following the usual rules, making use of our arrow trick, filling out the Punnett square for the sake of simplicity and completion, we find that all the offspring are all tall and yellow. So if you see that all the offspring are all tall and yellow, one should be able to anticipate or work backwards that there's no gene for smallness in that parent and no gene for greenness, and that's quite true, because the genotype for tallness in the parent is that it is big T, and the genotype for yellow colour is that it is big Y. So, putting this together, here are our results. If the parent is a heterozygous parent, and it crosses with a double recessive, we will get a ratio of 1:1:1:1, which is the cross we've carried out earlier on. But for a thinking cap question, we should also be able to perceive that if we don't know the genotype of the parent, that we can work backwards by looking at the offspring and determine it. So in a nutshell, if half the offspring are tall and yellow and the other half are small and yellow, because tall and small are showing up, as we've already said, the genotype for height must be big T, small t. Because both offspring are yellow, there's no gene for greenness in the genotype, so it has to be big Y. Again, same reasoning, there's no small appearing in the offspring, so the unknown genotype you can work backwards and determine that it has to be big T, and because yellow and green are making an appearance, you can work backwards and you can determine that the genotype of the unknown parent has to be big Y, small y. And again, the final result, that all the offspring are all tall and yellow, so you can work backwards and determine that the genotype of the parent for tallness had to be big T, and the genotype for yellow is big Y. Now that we have reached the end of our lesson, have we achieved our objective? Can you use the Punnett square technique to study the inheritance to the second filial generation, F2, of 2 unlinked traits?